Welcome back to Plantopia. Plantopia is the plant health podcast of the American Phytopathological Society, and I'm your host, Matt Kasson. I'm an associate professor of forest pathology and mycology at West Virginia University. The views and opinions expressed by our guests are their own and do not necessarily reflect the views of the host, the American Phytopathological Society, or any affiliated entity. This is the 7th episode of 2026 and the second of a 3-part series featuring 2026 APS Fellows. Today I welcome Dr. Rodrigo Almeida, Professor of Emerging Infectious Disease Ecology and the Hildebrand-Laumeister Chair in Plant Pathology in the Department of Environmental Science, Policy, and Management at the University of California Berkeley. Dr. Almeida completed a BS in Agronomy and MS in Entomology from the University of São Paulo. In 2002, he earned a PhD in Environmental Science, Policy, and Management from UC Berkeley, focusing on insect transmission of the bacterial plant pathogen Xylella fastidiosa. Dr. Almeida served as an assistant professor in the Department of Plant and Environmental Protection Sciences at the University of Hawaii, Manoa, from 2003 to 2005 before joining the faculty at UC Berkeley in 2006, where he is currently serving as full professor, interim faculty director of the Essig Museum of Entomology, and Hildebrand-Laumeister Chair in Plant Pathology. Dr. Almeida is nationally and internationally recognized for his research on insect-transmitted plant pathogens, notably bacterial and viral diseases of perennial crops. His multidisciplinary research uses a range of tools and approaches such as field and greenhouse studies, gene function research, and pathogen population genetics and evolution to answer fundamental questions in plant pathology regarding disease management and control strategies. Dr. Almeida research has yielded many biological, ecological, and evolutionary insights on the bacterial plant pathogen Xylella fastidiosa. This includes uncovering the role of host plant polysaccharides as cues for cell phenotypic changes that lead to vector acquisition, demonstrating that Xylella fastidiosa adhesions can be co-opted to compete with cells for adhesion sites, and developing efficient and simple protocols to knock out genes in a previously extremely challenging pathogen to perform functional research on. Dr. Almeida's research has resulted in over 140 peer-reviewed publications and numerous honors and awards, including serving as a Fulbright Research Scholar and Marie Curie Fellow, a AAAS Fellow, and a Fellow of the California Academy of Sciences. And finally, a UC Berkeley Grad Assembly Faculty Mentor Award. He currently serves as editor of Phytopathology and previously led a phytopathology special issue on Xylella fastidiosa. He also currently serves as a member of the United Nations FAO IAEA working group on a global program addressing food security and climate-smart agriculture. So welcome, Dr. Almeida. Congrats on being named a 2026 APS Fellow.
Yeah, thank you. Thank you. And it's a pleasure to be here with you.
Yeah, certainly. I assume you're being recognized primarily for your original research contributions, but given your professional service outputs, including work with the UN, the European Food Safety Authority, the Italian Ministry of Ag, it's clear that public service and outreach contributions are quite meaningful as well. Are you being recognized on those two fronts, or just really the original research?
I think it's mostly on the original research that a lot of the public service outreach that I've done has been in some ways mostly international on an ad hoc basis. And I would say a lot of it is related to the outbreaks of Xylella fastidiosa that have been emerging mostly in Europe.
Yeah. And we'll get into all that because I'm curious to hear about some of those projects that you've been involved with and publications that you put out. Now, you've been previously named a AAAS Fellow and a Cal Academy of Sciences Fellow. Clearly, you're not a stranger to being recognized for your scientific contributions. But how does it feel to be recognized by your plant pathology peers as an APS Fellow?
Yeah, I think it's actually quite special. It's one thing to be recognized by us for, in some ways, a more general type of academic contribution, but at the end of the day, while I identify in many disciplines or with many disciplines, I think plant pathology is ultimately my home, and to be recognized by APS is quite special.
Yeah, certainly. Your peers at APS are going to obviously recognize the impact of your work because they're working in that same space and they can see that you, among so many great scientists that are part of the society, you even stand out among them. So it feels extra special. So when did you first get involved with APS?
Oh, that's a long time ago.
Yeah.
I think I was a graduate student at the time, and
At Berkeley.
at Berkeley. I did my PhD at Berkeley, and in between plant pathology, entomology, and related disciplines. And I don't recall, but I probably attended a couple of meetings, and a way of not paying as much for registration was to become a member. And I do think that is the very beginning of my relationship with APS. And I do remember those early meetings as being very productive and exciting experiences.
Yeah, definitely. Now, you're originally from Brazil, right?
Correct.
And you earned a BS in agronomy and an MS in entomology there before coming to the US for your PhD studies, like we just discussed. What got you interested in agriculture and plants and insects to begin with?
Yeah, it's interesting enough. It's been at the very beginning of my career is just a sequence of failures and disappointments. In Brazil, it's a little bit different. When I finished high school, you essentially have these general exams, which are purely quantitative to get university. And I knew I liked biology, but I had no idea what I really liked to do. And becoming a physician was the obvious path of more security in some ways, career-wise. And I applied for, at the time, 3 medical schools, and I applied for what was at the time and may still be, I don't know, the best agronomy school in the country, which turned out to be 20 minutes walk from my parents. And I did not get in any of the medical schools, but I got in the agronomy school. And given that it was a good school and it was right there where I grew up, and there was no problem in trying it out. And so I enrolled and I started my degree in agronomy. I remember the first semester I tried to volunteer to work in the plant pathology department. I went there and the professor said, no, you have to take microbiology and plant pathology before you can volunteer as an undergrad. And I did that, but I remember disliking plant pathology in particular. I essentially had already passed the course before the final, so I stopped attending. Or in Brazil, your grade is from 0 to 10, and a 5 is a pass. So without attending and barely taking the final, I certainly had a mediocre grade.
Okay.
And so then I went there, and they interviewed a bunch of my peers, my cohort, for an internship. I went there, and they said, no, you didn't make it, which was quite disappointing, not being able to work for free.
Yeah.
And I think I did hang out around one lab for a little bit until this guy said, I have a colleague in entomology who's working with vectors of plant pathogens. And maybe you can go talk to him and at least he will stop hanging out around my lab. And I did that. And that's really how I started. And ironically, I did some work with insects very early on as an undergraduate in João Lopes's lab in entomology. Then I did my master's with him, which was probably the first master's in entomology that had a student that never worked with insects for the dissertation,
Yeah.
for the thesis. But yeah, so that's how I started, by not being able to, by being rejected twice by the plant pathology people.
Yeah. So coming back to the fact that you had this MS in entomology, moving forward, like thinking to your transition to your PhD, did you want to involve like an insect component? Because your PhD was on Xylella, fastidiosa, and insect vectors. So clearly there's this connection to your agronomy and entomology roots. But did you have like a thought that you wanted to integrate them into your PhD work, or is that just how it worked out?
It had always been integrated, even as an undergraduate and in my master's. I mean, my thesis did not include any of it, I think. I don't recall, but there were always insects around, and I was in an insect lab for sure, or a transmission lab. My PhD at Berkeley was with Sandy Purcell, was one of the main people when it comes to Xylella fastidiosa and phytoplasmas and other diseases. Sandy retired 20 years ago. And my interest there was to be trained as in the area of insect transmission of plant pathogens. That had always been my focus as a young scientist in Brazil. But then for my PhD, it was at this very intersection of these various disciplines. So working with Sandy was a natural sort of fit, studying transmission and related aspects. So I would say that my early career has been always focused on how insects transmit plant pathogens and related topics.
You're one of the world's experts on Xylella. This bacteria is the cause of several important plant diseases, including Pierce's disease of grapes, bacterial leaf scorch, phony peach disease, olive quick decline syndrome, citrus variegated chlorosis, among others. We know that Xylella fastidiosa can infect a wide range of plants, and across various publications, I see 300, I see 700 species, but it gets confusing, and I was hoping you can shine a light on some of this for our listeners, because what I read is that Xylella fastidiosa actually comprises at least 3 distinct subspecies with different geographic origins and host preferences. Is that right?
That's about right. As you mentioned, it can get complicated very quickly. So Xylella as a species, Xylella fastidiosa in particular, there's another recognized species, Taiwanensis, and there's some new work coming out of France that suggests that there might be other stuff out there too. But if we focus on Xylella fastidiosa, as a species, you can think of it as a generalist in some ways, that it goes to an ever-increasing number of host plants. You just have to keep in mind that it does not necessarily cause disease in all of those plants, right? The vast majority
Right.
of plants are maybe symptomatic, but we don't really know what's going on, or asymptomatic. A lot of those are positive or infected, rather than symptomatic diseases where you have outbreaks or problems of economic or landscape importance. Regardless, there are essentially 3 recognized subspecies. The current understanding is that, based on essentially biogeography and diversity, is that they originate from South, Central, and North America. And within those, you have subgroupings and a wide diversity of clades. Once you get in the weeds, you can have an idea of which plants are infected by which genotypes, let's call them. At the same time, part of the problem is you can have some plant species, for example, coffee, blueberry, olive, where you can have a multitude of very distinct genotypes that can infect those plants as well in the field. It has been really difficult because Xylella does not have a type 3 secretion system, so a lot of how we understand plant pathogen interactions for bacteria does not really exist in Xylella. And so we still don't really know what makes Xylella go to one plant and not another, for example. And it's an active area of research, but broadly speaking, I would say that we still don't know the answer to that.
So these 3 kind of subspecies that we're talking about, multiplex, which is the North American subspecies, or at least biogeographically originated in North America, subspecies fastidiosa, which has a Central American origin, and then the pauca, which it has South American origins. From what I was reading, it's the subspecies fastidiosa that's the one that's really been here in North America for the 300 years and is really associated with Pierce's disease of grapes. Is that correct?
Yes. What we are considering Central America and North America, and the boundaries of that from this perspective, are not particularly clear. And the reason for that is that between the US and essentially Brazil, there is very little work that has been done in terms of surveillance and diversity of Xylella, other than Costa Rica. So most of what we know about, again, in this context, broadly speaking, Central America, is everything between the US and Brazil in some ways. And
Oh, wow. That's a lot of space.
it's a lot of space. And in there, Costa Rica, we know a little bit of what's in there, and there's a lot of subspecies fastidiosa diversity. We have some data from Mexico. We have some data here and there from other countries, but it's not a lot. So anyway, let's just say that. But yes, fastidiosa in the United States, when it comes to Pierce's disease of grapevines, was introduced, at least we think it was introduced around 1750s. And it's been essentially, we think, one introduction somewhere in the US. We tend to think around somewhere between Texas and Florida at that point. And then from there, it has been introduced a couple of times into California. And then from California, it has moved back to Texas and other areas. So within the United States, there has been some moving of the pathogen that causes Pierce's disease, but it is considered an introduction. It's not considered to be native to the United States.
And a lot of this, like figuring out the timeline of introduction and the spread, This was highlighted in your 2025 current biology paper using some of the herbarium specimens. Is that correct?
Yeah, that's correct. That's actually all credit here goes to two students in my lab, Monica Donegan and Alexandra Kahn, and collaborators from France who were in a team in La Réunion led by Adrien Rio. And Adrien was a specialist or is a specialist in using herbaria samples for reconstructing plant disease outbreaks. And we had a very tiny grant between the US and French embassy at the time for them to come over for a couple of weeks and search for Xylella samples in herbaria at Berkeley and at Davis, which it's a little bit of a needle in a haystack because Xylella Pierce's disease symptoms make the plants look scorched and just not good herbaria samples.
You wouldn't put them on press sheets because they would be ugly samples, right?
Yeah, exactly. And so they came here. It was Adrien and Nathalie from the French team and Monica and Alexandra from our group. And they went through herbaria samples and they came across one sample that said Anaheim disease. And that in the early 1900s was one of the names that, for Pierce's disease at the time. Pierce's disease emerged in the 40s, 50s as a name for the disease. Anyway, yeah. So that one sample, they collected one sample or found one sample from 1906 from Modesto, California. And that sample turned out to be positive and they were able to sequence the whole genome out of it. Then you just go and you do your phylogenetics and molecular dating, and that shifted the introduction to around 1750s or something, which was really cool work that they did.
Yeah. This question may be a little naive, but if your earliest herbarium specimen and genome, bacterial genome, and I don't know, did you get a full genome from that, is 1906, how are you able to reconstruct its introduction back to the 1700s?
Yeah, so they were able to sequence a full genome, which was very exciting. And from a gene gain and loss perspective, it's actually pretty boring. It does not have anything particularly exciting. But you can then use a historical genome, in this case, 120-year-old genome, you can place it in a phylogenetic context and you can start looking at substitution rates. It's essentially the number of substitutions accumulated over time. And if your data are good enough and satisfy assumptions, you can actually then generate essentially a molecular clock, right? The number of substitutions that you have accumulated over a period of time.
Yeah.
And then you just go to your phylogeny and you see what's the node that is related to the introduction itself. So that's what they did, and it seems pretty reliable. And I think that at the end of the day, if we can find more of these old herbaria samples that have Xylella infections, that would be really helpful in getting an even better idea of the history of these outbreaks.
Absolutely. And I think you also bring up another point that maybe wasn't necessarily on my question sheet when we started this, is that here we are, this is published in 2025. Presumably the work may have started a couple years before that, but the techniques and tools we used to probe these historic specimens occurred within the last 5 to 10 years. And it really tells us that holding on to these herbarium specimens is really important for making scientific discoveries and remains as such. I know there's been a lot of pushback to get rid of historic herbarium collections and fund these kinds of things, but clearly even this work that you published in 2025 in Current Biology shows that these specimens are really important for reconstructing the history of these disease epidemics.
Yes, that's absolutely correct. I tend to think of these as data-rich time capsules that store so much information, some of which we can start taking advantage of or looking into, such as pathogen sequences, for example. But there's potentially a lot more that we have never thought about. And once they're gone, or if we can't store them properly and degrade it over time, they're lost, right? So there's so much potential there to store, not to store, but so much potential for research in the future to reconstruct epidemics or understand pathogen evolution, for example, in our case. And there is a need to figure out how to preserve this material. The same thing is true for cultures, right? Introduced pathogens, emerging diseases, having early cases stored in perpetuity so that people can 10, 20, 50 years from today, 100 years from today, go back to those and do something with them that might be actually really helpful in the future.
Are there actual examples of Xylella fastidiosa showing up accidentally in plant genome sequencing projects from herbarium specimens that have come across your desk that have been useful for your work, or hints to them being in some of these maybe sequenced plant specimens from other herbaria around the globe?
Yeah, so not from herbaria necessarily, but a French group from Angers in France, they looked up into sort of plant sequences, right? And they were able to find some Xylella like or putative or potential new species of Xylella in DNA sequences from Asia. I'm assuming they're continuing that work. It was a report on here. We found these sequences in some crops from Asia. But yeah, I think that there is a little potential for trying to learn more about what's out there from unexpected sources of information or unexpected sources of data, such as genome sequences from unrelated crops or things that you're not looking for that, but maybe there is some information in there.
Yeah. I want to keep on the topic of these subspecies within Fastidiosa because some other things caught my attention. As reading through. I saw mention of some other subspecies, Sandri and Tashki and Morris. And in your 2018 annual review of phytopathology article, you mentioned intersubspecies genetic introgression between Fastidiosa and multiplex, and that's what we call Morris. Is that correct?
Yeah, that's correct. So to go back a little bit, early work done by folks at UC Riverside showed that with a multi-locus sequence type in MLST, when you sequence 7 genes, housekeeping genes to just essentially for genetic typing, they found some evidence of recombination, homologous recombination in Xylella, which led our group at the time to look into it from a biological perspective, and we showed that Xylella is naturally competent. As it turns out, Xylella recombines a lot. Its natural competency is extremely efficient. And you look at genomes and recombination versus mutation rates, and recombination rates are much higher than mutation rates, in fact. So there is a lot of recombination going on. The proposed subspecies Morus may or may not be a subspecies. And in fact, it goes back to the beginning of our conversation. And I would say that Xylella is it's a taxon that desperately needs taxonomists to go into work on it a little bit. Not my job, but I would be happy someone could go in there and sort things out a little bit more.
Yeah.
But Morus, as proposed, it's mostly fastidiosa, I think, with up to 10% of multiplex sequences in there. But there is a lot of recombination in Xylella that goes back and forth. A lot of it is within subspecies, but there is also a lot of evidence between subspecies recombination. And I think that it does complicate a little bit the taxonomy and some of these details as to, is it a subspecies? Is it not a subspecies? Do you use the core genome? Do you use the entire genome? And yeah, it just all complicates things quite a bit.
And I know obviously having the full genomes and be able to do genome-wide phylogenies, and that is the gold standard. There's some complicating factors here for our listeners that may not be super familiar with Xylella fastidiosa. Its species name is fastidiosa, which means it's fastidious, or simply put, it has complex nutritional requirements, and it makes it difficult to culture on artificial media. In fact, if I remember from my plant pathology PhD first year, we learned that it took 80 years to figure out how to first culture it. And I remember there was a publication, 1980, in phytopathology by Davis et al., where they came up with an isolation media for Pierce's disease bacterium. Was that the first successful culturing back in the 80s or late 70s, early 80s?
It was late 70s. Yeah, Mike Davis was a graduate student at Berkeley. And as I recall, he spent a few months trying to culture Xylella. And eventually he was able to do it. To his credit, a lot of people had tried to do it in the past and they couldn't do it. But yeah, so Mike Davis was the first person to be able to culture it. Then he developed a few different media. They've been successful. Other people have done more on that front. But yes, generally speaking, it's not particularly easy bacterium to culture. There's variation based on genotypes as well. The one causes disease in grapevines in California. You can grow it within a week. You start seeing colonies. But others, such as citrus in Brazil or olive in Italy, for example, it will take you 3 weeks to start seeing tiny little colonies, for example.
Yeah.
So it's not necessarily a very easy one to culture. And that may have led to a smaller than desirable number of genomes out there.
Yeah, I was going to ask if that's the bottleneck in high throughput genome sequencing of Xylella is the fastidious nature.
I think that in the US in particular, not so much, because the one from grapevines can be cultured fairly easily. And some other crops, you can do it from almonds, for example. But in Europe in particular, and for other plants in the United States now that there is a lot more interest. So essentially in the last 10 years, yes, there's been a lot of interest in trying to get more of these strains in culture, but a lot of them are really hard to essentially to isolate.
Yeah.
And so it is a restriction today because we're not working only with grapevines, essentially, which is of the disease systems, maybe the most tractable one from that perspective.
Sure. So as a forest pathologist myself, I'm obviously really interested in shade trees and forest trees, and presumably multiplex, the subspecies multiplex is the one that is involved with bacterial leaf scorch, or predominantly involved with bacterial leaf scorch of various oaks and other shade trees. Is that correct?
As we understand it, yes.
Okay. And that one is not as easy to culture as the fastidiosa subspecies is.
Again, yes, generally speaking.
Okay.
So the fastidiosa that infects grapevines in the US is fairly easy to culture as far as Xylella goes. If you go to Costa Rica, a fastidiosa that infects coffee and other plants in Costa Rica, it's not as easy to grow, for example. Then you go to multiplex. The multiplex that infects almonds in California is fairly easy to culture. The multiplex that infects urban suburban trees in Southern California, for example, is very difficult to culture, as is a lot of multiplex that infects oaks, for example, as you mentioned. So there is a fair bit of variation. Multiplex tends to grow slower. It tends to be more difficult to culture. But there are some specific genotypes that I guess you can culture a little bit more easily.
Now, which of the subspecies was implicated in the olive die-off's immortality in Italy? And was that the Pauca?
Yes. So that it's a genotype of Pauca, but it's not nested within the traditional Pauca group from South America. It's a sister clade, and that's connected to Costa Rica, although it may be originally from Costa Rica, but if you only have data from Costa Rica for Central America, then it has to be Costa Rica.
Yeah, I see this. Further complicating this, which we'd love to bring our listeners along for a complicated voyage, I'm thinking about the distinct, maybe unique insect vectors for each of these. So with these different subspecies, do we see a lot of overlapping insect vectors, or are they unique to each?
Yeah, this is the one cool thing about Xylella that it's sometimes hard for some people to accept if they're not familiar with the system. There is no evidence of vector species Xylella genotype specificity.
Okay.
Essentially meaning that any xylem sap sucking insect can transmit any genotype of Xylella fastidiosa within reason. But so in the Americas, sharpshooter leafhoppers it's a subfamily. Those are the main vectors of Xylella in the Americas, from all the way from Brazil to the US. And you can get insects from that group to transmit any genotype of Xylella that has been tested. This does not mean that they do it efficiently, just that they can do it, right? It's a yes or no sort of question.
It's not stylet-borne though, right?
It's not stylet-borne, it's foregut-borne. So essentially, it colonizes the cuticular lining of the mouthparts, but past the stylet. So the stylets are essentially what goes into the plant, right? Like a little straw. And they diverge into this structure called the precibarium, which is a narrow canal before the cibarium. And the cibarium turns out to be a pump. So you
Okay.
can think of just a pumping system. And Xylella tends to occur in this precibarial region and a little bit in some areas of the cibarium itself. But so that's the region that's colonized by Xylella. So most insects have hundreds, a few thousand cells, but there's limited space
Yeah.
colonization and a small number of cells overall.
And we're talking about Hemiptera here, which have a nymph immature stage. And I read that this is semi-persistent in the nymph stage, but it's persistent in the adult stage. Is that correct? Where it's because they have to go through molts in the nymphal stage, they'll basically lose the bacterium, but then they have to regain it. Is that accurate?
So the biology is accurate, the terminology is not.
Okay.
So the biology is accurate, which is what, at the end of the day, is what really matters. So the cuticular lining of the foregut is of ectodermic origin. Essentially, it's just like the exoskeleton. When the insect molts, it changes its exoskeleton, essentially. And the foregut goes out with it. And if you have a Xylella colonization of the cuticular lining, that goes away with the rest of the exoskeleton. It's not semi-persistent in the sense that for semi-persistent transmission, the pathogen gets lost over time essentially from the mouth parts. It's the case of some viruses, for example. So in a matter of generally days, the insects lose the infectivity. So the viruses are no longer where they should be for infection, for inoculation to occur. So it's persistent. If the nymphs never molted, they would continue to have the infection.
Got it. That makes sense. So one of the things I noticed in your APS Fellow profile was that you developed an extremely efficient and simple protocol to knock out genes in a previously challenging pathogen. We're talking about Xylella fastidiosa. Does that have something to do with culturing it, or how did you accomplish that? Like, how did you make something so efficient and simple that was otherwise really challenging to work with?
Yes, that is correct. Although I will give pretty much all the credit to Leonardo de la Fuente at Auburn University and his students for actually developing the system. So essentially, when we found that Xylella was happy picking up DNA from the environment, and that could be done very efficiently, both linear DNA, plasmids, Leo de la Fuente's students, we contributed to that work, but they really led all that work. They developed a system where essentially you can give a plasmid or linear DNA to Xylella with it's essentially a little construct, and you let the cells grow for a few days, and you spike your culture with your construct, and you just plate the cells in mycin, a medium with kind of mycin. And just like that, you get hundreds of knockouts, which is really cool. My dream is to one day really be able to scale this so you can knock out pretty much every gene and see how that works.
Yeah.
But we've tried and we have not been able to get the funding for it yet.
I wish you luck in getting that, of course. In your molecular plant perspectives paper in 2020, you say genomic data are beginning to be used for an array of evolutionary and ecological questions that were previously impossible to address, including the use of genome sequencing to provide sufficient genetic resolution to study pathogen dispersal pathways. We talked a little bit about your current biology paper and how you're able to do that. But you also acknowledge in that paper that genetic diversity can be found within regions, orchards, trees, and even branches of studied agronomic crop species. Yet we have, at the same time, have little insight into what's happening in natural ecosystems. Now, that paper was 2020. Do you think that we've, in the last 5 years, have started to fill the gaps of what's happening with Xylella in these natural ecosystems? And what kind of information has that provided, if so?
Yes, I think so, particularly in Europe, but a little bit also in California and Eastern US. I would say that the most of the information has been on increasing the list of potential hosts or plants that are infected with Xylella. I think in Europe, there has also been a lot of interest in trying to understand new outbreaks and how they may impact non-crop species, so essentially landscape trees or forest environments. It's not completely clear how that has or if it has led to management practices in Europe necessarily or the US, but I think there's a lot more knowledge and a lot more interest in regards to what plants may or may not be infected, rates of disease, how much of a problem you may or may not have in environments where there are not your traditional crop focused type of disease like much of the Xylella research in the United States.
Yeah. Having worked on verticillium wilt for my PhD, I see some parallels between verticillium wilt and Xylella, because verticillium dahlia, for example, has this really expansive host range list. But like when you start to get into the MLSTs and genome level resolution and that, you start to see, okay, maybe it's not as generalist as we thought. It has these different clades and things like that. The other thing that we're starting to understand with verticillium, and you brought this up with Xylella, is that there's a lot of hosts that are asymptomatic hosts. It's there, it's present, you can isolate it out, you can detect it using molecular techniques, but it's not really having an outward effect on yields or health of the plant, or at least outward health of the plant. So I think it's challenging, because that could mean that you could sample everything and find it many places. Was only limited funding to do that.
Yeah, no, I think that's a good point. I would say that in the case of Xylella in particular, we are just at the very tip of the iceberg. There is a student in my group, Patrick Lee, who's looking at urban suburban trees, so essentially landscaping trees in Southern California, San Diego, Los Angeles County. And he has found that lots of trees there are infected with Xylella. He's reporting a lot of new and unreported diversity from these trees. Just driving around San Diego County, for example, looking at olive trees that he's like, oh, okay, look, that looks like it has some scorch symptoms. I'll take some samples. So not completely unbiased in the sense of where he is, but biased in the sense he's driving around and collecting samples from random trees in many ways. If they have any sort of scorch symptom, he has been able to find over 80% or more infected trees of these samples that he took, which is just an absurdly high rate of infection, right? If 80% of olive trees in Southern California that have some scorch are infected with Xylella, it just shows that you have very high rates of infection out there. I want to come back for a second to this question of diversity and getting to trees or regions, and just mention some work we've been doing. We have about 200 genomes of Xylella from grapevines in California, to Southern California to Northern California, and a student in the lab, Monica Donegan, who's just graduating now. Monica did a little bit of a landscape genomics, genome temperature association study, and she's been able to identify a few loci that correlate with temperature, summer and winter temperature, and she actually knocked out some of those genes. And she's found that they have a phenotype correlated to aggressiveness, but also winter survivorship of the pathogen. The point here being that we always thought, when I was a student, that there was one strain or one genotype, whatever that would mean, causing disease in grapevines in California. Now we know that each region has its own genotype, but more than that, we also have a fair bit of evidence that this has accumulated, demonstrating that there's some evidence of local adaptation, and that you can find that evidence of local adaptation experimentally, but we also can find in the genome, all the way down to knocking out genes and seeing a phenotype that correlated to climate. There's a huge amount of diversity there that we tend to, I wouldn't say ignore, but think that it doesn't matter from an applied perspective, at least from my view. And when we think about trees or animals, for example, we're like, of course, animals in Southern California are going to be different from animals in Northern California in terms of what they do and what they look like and their physiology. And the same thing is true for trees, for example, right? But then we go to plant pathogens and they're all the same. And which obviously is not correct. The question is, does it matter? Can we show it? Those are different types of questions, right? But
yeah, more importantly, as a follow-up to that, because I think I think listeners are interested in connecting the dots of how these discoveries of these regional specific clades or whatever, how does that influence management of the disease in those different areas?
Yeah, that's an interesting question, isn't it? So the obvious one for us in California in particular is where you go, for example, table grapes may not be the same area where you grow wine grapes, for example. So in coastal California, you grow pretty much only wine grapes. In the Central Valley, you grow both, as an example, right? With some variation depending on the region and whatnot. But if you're thinking about breeding, for example, for varieties in coastal California, maybe you should get strains that occur in coastal California, for example. And if you're thinking about breeding in the Central Valley, maybe you should use strains from the Central Valley. We do have data showing that they are different in terms of aggressiveness and whatnot. We also do have data showing that there is variation in terms of total responses. So that's one easy way of thinking about it. The other one is risk. So as we mentioned earlier, I do talk to folks in Europe quite a bit about risk. And the question here being, should risk models, in terms of what are the risks of introduction, should they consider the entire range of Pierce's disease in the United States, for example, in terms of temperature and geography and whatnot? Or should they consider the risk based on where the strains come from, for example? At the end of the day, you would assume that an introduced pathogen would adapt over time to local conditions, right? But at least in the beginning stages of an outbreak, does it matter where they come from? If they come from Southern California or Northern California, does that impact your risk, at least in the short term? And I have no idea if it does, but these are ways that you can think about it from a more applied perspective. Why should we care about this variation that it's out there? And again, I think one of the challenges one, you have to show that it's out there. Two, what does it actually mean? Can you come up with experiments that demonstrate that you have meaningful differences, and do they matter at the end of the day?
One of the things that comes up just about every interview I do for this podcast is we have a particular researcher that works in a specific system, and they've made a lot of progress in whether they're an APS Fellow or an academic or a government researcher. We bring up the fact that diseases don't occur in a vacuum. There's other pressures outside that pathogen-host interaction. Grape, for example, has plenty of diseases outside of Pierce's disease. In your efforts to understand Pierce's disease and how it impacts the grape production in California, are there other pathogens that are proving to be more challenging to manage or that you have to consider when you're thinking about managing Pierce's disease, whether it be like a gray mold or powdery mildew, like these other oomycetes that might impact grape, something like that? Are they more challenging than Xylella, or does Xylella represent one of the largest challenges to grape growers in California?
It's an interesting question. So there are a couple of things to consider. One is when you have a Xylella infection, it is either your plant's not going to produce very much or anything within 2-3 years in a colder climate, or it may die in 2-3 years in a warm climate, depending on variety and the condition. So things like powdery mildew, for example, become very secondary in terms of what kind of problem you have to deal with. Essentially, if you have a Xylella infection, you should be roguing your vines, and that's it, as fast as you can, or as fast as you're comfortable with based on the symptoms. The other thing I would say is that Xylella outbreaks in California, there are currently essentially two very different types of outbreaks. One is in Southern California, driven by an invasive insect, the glassy winged sharpshooter, which is an outbreak that has gotten a lot of press. Has been a very important outbreak and started in the late 90s, early 2000s. And the ecology of that is a little bit different from let's call it the traditional Pierce's disease in California, which is driven by a native insect, the blue-brain sharpshooter, in coastal California. And the glassy winged outbreak has been really managed by vector suppression, removal of infected plants. These are area-wide programs between the California Department of Food and Agriculture, CDFA, and USDA managing these efforts. These are large efforts, and they've been fairly successful. But if you go to the native vector situation in coastal California, in those, disease occurs generally in sort of cyclical outbreaks every 15 or 20 years. So the last big one was around 2013, 14, 15 in coastal California. It was pretty devastating in some locations, some vineyards with 20, 30% losses, which for a vineyard is very significant.
Yeah.
But you go to Napa right now, for example, Napa Valley, where traditionally you can always find some Pierce's disease. And you go look for it, you can't really find it. So the last 10 years, essentially, in Napa, there has been very little disease. The reasons for that, we don't understand. Maybe people were very good at removing the sources of inoculum, or maybe we're just waiting a few more years for another cyclical outbreak. But so I wouldn't say that other diseases impact Xylella as much in terms of management, because you see a sick plant, you should remove it. Kind of end of story in some ways. And for the most part, it's very cyclical. And by the time the new outbreak shows up, vineyard managers have in many ways been replaced by younger people, and I forgot about it. And then you have an outbreak, and by the time they understand what's going on, it may be a little bit too late. So extension colleagues are trying to develop monitoring programs, so they're a little bit more aggressive. If you have a new outbreak emerging, before waiting for things to get bad, they can be more proactive and respond quicker to the risk of the outbreak really emerging and causing a lot of problem.
Now, I read that hemipteran vectors are often called the only means of natural dissemination. And I assume that means long-range dissemination, because can't intraspecific root grafts occur between adjacent plants to allow for root graft transmission, or does that not happen in grape?
As far as I know, grapevines don't root graft.
Oh, okay.
I might be wrong, but as far as I know, that's the case.
And perhaps even if they did, spacing technique could be such that would limit that or that. Because I know even in managing root graft transmission of vascular wilt fungi, for example, just thinking strategically about spacing could really minimize that from occurring. One last thing I wanted to ask you about, because we're quickly running out of time, is in the East here, we have a new invasive pest called spotted lanternfly. And I do know that it does enjoy grapes. And in fact, it's presented some challenges, I think, in the Finger Lakes region. I don't know if it's up in the juice-producing areas of Pennsylvania, but this is a plant hopper. And I didn't know if it actually could successfully vector a Xylella or not. Maybe it can't, but it's something that came to mind as you were talking about the fact that there's not as much specificity as with some pathogen vector interactions.
That is correct. There is not a whole lot of specificity as long as you're within certain groups of hemipterans. Okay. So this particular insect is in another group. It's another family, Fulgorid. There is no evidence of any Fulgorid transmitting any Xylella.
That's probably a good thing, given how invasive they are here in the East. And I don't know how far west they've gotten, but they're pretty bad here in the Mid-Atlantic and have been for a while now.
Yeah, I think at the end of the day, never say never. But until people do plenty of work and conclusively demonstrate that it's a vector, from my perspective, I would say that it's not expected to be a vector.
Unfortunately, we run out of time, but I've enjoyed talking to you about your research and some of the work that you and members of your lab have been involved with over the past several decades. So certainly appreciate you taking time out of your schedule to meet with us.
No, it has been a pleasure talking to you, Matt.
You've just heard from Dr. Rodrigo Almeida, Professor of Emerging Infectious Disease Ecology in the Hildebrand-Laumeister Chair in Plant Pathology in the Department of Environmental Science, Policy, and Management at the University of California Berkeley. Be sure to check out the show notes for links to Dr. Almeida's APS Fellow profile and his research program at UC Berkeley. I'm Matt Kasson, host of Plantopia. Thanks for listening.